Hello everyone. Today, we embark on a journey through the fascinating history of cell biology and physiology. Together, we will explore pivotal discoveries that have shaped our understanding of life at the microscopic and physiological levels. Let's dive in. Our exploration spans centuries, starting with the understanding of blood circulation in 1628. We navigate through key milestones, such as the discovery of the cell in 1665 and the formulation of the cell theory in the 19th century. Later, we delve into the world of organelles, which was discovered in the late 19th century. Continuing our journey, we reach 1948, marked by the discovery of the Calvin cycle. Finally, we explore profound insights into nerve impulse transmission in the 1950s and the mechanisms of hormone action in the 1960s. Let's embark on this historical voyage, starting with the significance of these milestones. Cell biology and physiology constitute the foundation of our understanding of life processes. Through the study of cells and their functions, we gain insights into the fundamental mechanisms that propel living organisms. This knowledge is vital for advancements in medicine, biotechnology, and our overall comprehension of the natural world. Our journey begins with William Harvey. Through observations and experiments, Harvey discovered that blood circulates throughout the body. His experimental work was a trigger for the development of modern life science. De Motu Cordis, published in 1628, is a groundbreaking work that summarizes Harvey's ideas on the circulatory system. Translated as an anatomical exercise on the motion of the heart and blood in living beings, this seminal text systematically presents evidence to support Harvey's discovery of blood circulation, laying the foundation for modern physiology and cardiology. The image on the right depicts an experiment from the book. Then comes Robert Hooke. Using a microscope he invented, Hooke observed various minerals, animals, and plants. He discovered that cork had many small honeycomb-shaped structures, which he named cells for the first time. The invention of the microscope significantly contributed to the study of cells and living organisms. The image on the right shows his microscope. Micrographia is a seminal work in the history of microscopy, authored by Robert Hooke and published in 1665. In this groundbreaking book, Hooke documented his microscopic observations, revealing a previously unseen world of intricate details, including the first recorded observations of cells. The image on the left is the title page of Micrographia, while the image on the right is Hooke's observation of a thin slice of cork. The 19th century brought us the formulation of cell theory by Matthias Jakob Schleiden, Theodor Schwann, and Rudolf Virchow. Schleiden, in 1838, argued for the plant cell theory, proposing that plants are made up of cells after observing each part of the plant under a microscope. Schwann, in 1839, put forward the animal cell theory, suggesting that animals, like plants, are made up of cells. Virchow, in 1855, argued that all cells come from pre-existing cells and established the cell theory. Cell theory revealed that the basic unit of all living organisms is cells, promoting research on cells. The image on the right is an illustration of Virchow's cell theory. This theory unified our understanding. All living organisms are composed of cells. Cells are the basic units of life, and every cell comes from a pre-existing cell. As technology advanced in the late 19th century, so did our understanding of cellular complexity. Thanks to developments in microscope production technology and cell staining techniques, organelles such as mitochondria, chloroplasts, endoplasmic reticulum, and the Golgi apparatus were discovered. Chromosomes were also identified during this period. The image is a diagram of a typical animal cell, with the organelles labeled on the right. Fast forward to the mid-20th century, where scientists like Melvin Calvin and Andrew Benson unraveled the intricacies of the Calvin cycle. This discovery significantly advanced our understanding of how cells harness energy. Calvin and Benson used the radioactive isotope carbon-14, along with chromatography, to discover the pathway of glucose synthesis from carbon dioxide. The image illustrates the 3 stages of the Calvin cycle. Now, let's delve into the fascinating world of nerve impulses, where the work of Alan Hodgkin and Andrew Huxley made significant contributions. In the 1950s, these 2 scientists unveiled the intricate mechanisms governing the transmission of nerve signals. Hodgkin and Huxley investigated the initiation of action potentials, depending on the membrane permeability of ions. Through meticulous experiments, they elucidated the dynamics of ion movements across neuronal membranes. When a nerve impulse causes sodium ions to enter the cell, it becomes depolarized. The accompanying image illustrates this depolarization of the membrane. Now, let's explore the groundbreaking contributions of Earl W. Sutherland. In the 1960s, Sutherland contributed to the development of physiology by identifying the process of hormone action in target cells. Sutherland's research illuminated the cyclic AMP signaling pathway, revealing how hormones transmit signals within cells. His discoveries revolutionized our comprehension of cellular communication and opened new avenues in the fields of physiology and medicine. The diagram on the right displays a skeletal formula of adrenaline, a hormone studied by Sutherland. In closing, we've traversed centuries of scientific inquiry, from the humble observations of cells to unraveling the intricate dance of hormones. The history of cell biology and physiology is a testament to human curiosity and the relentless pursuit of knowledge. Thank you for joining me on this journey through the history of cell biology and physiology. May our curiosity continue to drive us toward new frontiers of understanding. Have a wonderful day. Thanks for watching. Don't forget to like and subscribe.